Hello, welcome back to the second part of RNA sequencing data analysis workflow tutorial. In this video, I will discuss about the different available databases where you can freely download sequencing data and perform sequencing data analysis workflows. First of all, I would like to talk about the NCBI. As you know that on NCBI, you can download different research papers and you can also look for different kind of data sets. For example, you can download sequencing data. So first thing is NCBI. NCBI is very popular and powerful database where you can find not only the data but also the different kind of research papers. Personally, I like NCBI because in NCBI, when you are new to bioinformatics, you don't know what to do and what tools you need to use for each step and where to get the data. NCBI is a place where you can find different kind of research paper, the older one, and if you find, for example, a research paper where they have performed RNA sequencing data analysis, they will provide you the data, like you can get the data from there, and for each step they have the paper. Like in paper you can read about each step, the tools they use and what were the options they use. You can just replicate their results and see if you have got the same result or different results. So this can be helpful for you if you are new to bioinformatics and you want to just make your hands dirty with some kind of data and sequencing workflows, then NCBI is good. Second database I would like to talk about is ENA, which is European Nucleotide Archive. This is also very famous and I like it because I normally what I do is my first preference is NCBI and second preference is ENA. Both have the same thing, like you can get the data as well as the paper. Paper is kind of a guideline. You can use the paper to download different kind of data, first thing, and secondly, for the same data you can get the pipeline, like if they have performed data analysis, what steps they used and what tools they use. So you can really get into the workflow and you can analyze it. So now I will show you how you can download the data, and this data I am going to use for my own data analysis for this video. Welcome back. Now in this part I will show you how you can download data from NCBI and ENA. Both databases are user friendly and I really like both of them, and let's explore one of them. First is NCBI. First of all, you just need to go to the Google and say NCBI and you will get this first link of NCBI. You just need to go to the link and this is the appearance or interference of NCBI. NCBI is very powerful. You can find all kind of paper and different kind of data sets. Right now I'm gonna demonstrate how you can use the GEO database. GEO is specialized for sequencing data. You can find more data, but they more specifically have sequencing data. So let's say here I type RNA-Seq and Homo sapien. I want to work with the Homo sapiens. You can write any gene, anything you want to work. You can write the name of the species. You can write the name of the technique. For example, you can write whole genome sequencing. You can write exome. You can write RNA sequencing, single cell RNA sequencing. You can use any keyword and you will get the result, but the result will be like a really big result. For example, if I click search, I will get the result like this, and it is almost like 0.1 million 17,448. So these are all paper where they use RNA sequencing. So okay, let's say we want to we open the first link where they have done retrospective identification of cell intrinsic factor that mark pluripotency potential in the rare somatic cells, and they use RNA sequencing for this. So if you open this, you will get this information. Like here you have a lot of information. The first thing is what is the status, like when it was published. It was published like just yesterday, and the title was like, this is the title, and then you have the experiment type, expression profiling by high throughput sequencing. And then what they have done, and this is really interesting, I really like this part, because here you can see they have talked about different things about the experiment. They talk about what they have done, so you don't have to read the whole paper. When you are trying to select a data set, you can just read this information. If it sounds interesting to you, you can go ahead and use it. If you don't want, you can switch to next one. So here I want to discuss one thing. When we do the sequencing, there are two types of sequencing. One is pair end, second is single end. So pair end is when we do the sequencing from the both end. Single end is when we do the sequencing from the one end of nucleotide stand. So it is pretty simple concept, but you will find it whenever you are searching for data. You will find two kind of data. One is pair end sequencing, second is single end reads. So you always remember like what kind of data do you want to use. I'm gonna use the pair end sequencing data for this tutorial. So let's say I'm not happy with this data, so I will open the second link, and in the second link we have this information. And after reading all this information, what we can do, we can simply jump here. This geo number, like the serial number, is very important, and just down to this serial number you can find SRA. SRA is a toolkit that you can use to download the data. So best thing is you install the SRA kit and then copy this number and just put this number in SRA toolkit. You will be able to download the data. So this is the one way, and this paper gives you all the information about all the steps, starting from the experiment design until the interpretation of the information from the data analysis part. And second database I want to talk is European Nucleotide Archive. I really like this. I will tell you why I normally prefer to use ENA. So first thing, this is for example your database, and this is the interference. Here you can see different tabs and tools. So we are going to search data. So we click here, and once we click here, after some time it will show us the search tab. Okay, so here we can search anything. For example, I want to search RNA-Seq data. I type RNA-Seq data and search, and you will get a lot of options. So you have to select one of these. So what I will recommend you whenever you are searching for data, so you will prefer this SR projects. There are like view all 12,000. So click here and you get the list of all the projects where they have the data from RNA sequencing. Here you can see, for example, the first project session number is this, and then the description is the RNA sequencing data. Second session number is for E. coli RNA sequencing data. So here you can really get the information that you are really interested in. So for example, we go to the first and second. So the first, here we have the information. So this is the data. First is the information like where this data came from. RNA sequencing data of SMAX1 mutant was wild type. So in this experiment, what they have done is they compare wild type and the mutant gene SMAX1. This is a paired end sequencing, like you have the two reads. Here you can see the two reads. So what you can do is, like, when you are on this page, you can just click on this option, see all the option, like what information do you want to see. I have selected these options, and you can use these options. Like I really wanted to see sample title, so I put it there. And then I also want to see the size, so I selected the file size, and also other kind of information according to my requirement of analysis. But you can do the selection according to your own choice or own requirement, so you are free to do that. Okay, so this is the information. So the best thing that I really like about this tool is what you need to do. You need to select all these reads from first to last if you want to download all. So I want to download all of the files, so I have selected all. So then I am going to click on to download the script. If you click here, you get a script and you can open this script here. What is the benefit of downloading it on the script? I mean, I always use this script to download the data. I normally don't prefer to install SRA toolkit. The reason is like it is more easy. I just need write like one code line. Once you have downloaded this script, what you can do is very simple. You can just copy the first line of the code. I mean, you haven't write this code, and you can just use this pre-assembled code for downloading the data. So first line I have to copy, and then I have to go to my terminal, and on my terminal I just copy this and then click enter, and then it will start downloading the data on my system. This is how you can download this data on your system, and this is pretty simple. I will show you how you can run this whole file on your system, but I think the data is like around 20 GB and it will take a lot of time, so I will prefer to download each file one by one the way I have shown you. So next step, I will show you how you can set up the environment, the system for your data analysis part or workflow. I would like to tell you one thing. This video, if you follow all the videos, it will take some time for you to learn everything, and it took me some time to learn everything actually. Be consistent in learning, and all these things will be nothing for you. Honestly telling you, when I started learning, it took me some time, and no, the same thing that took me to learn like 3-4 hours, now I can do in 4 minutes. That's why I really like this field. You spend the time to learn something and then you can do it like quickly, really quickly. So in the next part, we will do the system setup where we install a different software that we need to do the analysis. And after that, we will officially start our analysis. So we are spending a lot of time on preparation, but it is required. Thank you very much for watching the video. I will come back soon with next video where we will do the system setup together. Bye bye. Take care.